Hi everyone, welcome to today's Precision Systems Teatime Webinar Series. Today with us there is Dr. Hasan Issa from the University of Goethe, and he's going to talk about LNPs, therapeutic for acute myeloid leukemia. Before jump into the presentation, I would like to introduce about our Europe team. We are a team of 5 people, 2 RSMs and 3 scientists. Jürgen, he is based in Germany. Suha, she is based in the UK. Edward is based in Scotland, and Martin based in Germany, and myself in France. Please do feel free to reach us out if you have any questions. Thanks a lot again, and over to you, Hasan.
Thank you, Sarah, for the introduction. And it gives me an honor to present our work for you today. So my name is Hasan Issa. I'm a postdoc at Goethe University under Professor Klusman group, who is now the director of the pediatric clinic here at Frankfurt. So I will talk about 2 projects that I have been doing for the last 6 to 7 years. These projects included my PhD project, as well as one part of the postdoc project that I have done with Professor Klusman. So the projects are based in developing therapeutic LNPs to cure pediatric acute myeloid leukemia. In general, children myeloid leukemia is caused by different genetic aberrations and oncogenic event that hit the hematopoietic stem cells at different stages of differentiation or on the long-term stem cells, which disturb the production of mature blood cells and lead to accumulation of undifferentiated progenitors with enhanced self-renewal. This is how we define acute myeloid leukemia in children. Now, there are different genetic aberrations that might cause that. These mutations could be due to a translocation or a mutation that hit transcription factors that control self-renewal, differentiation, or drug resistance. And all of these enhance the survival and the clonogenic effect of the progenitors and block the differentiation. Based on these categories and the group of aberrations or genomic aberrations, children will have different treatment based on those, and these also correspond to the clinical outcome. So some of those genetic aberrations are associated with good prognosis, and others are associated with very inferior prognosis and poor clinical outcome. So in general, as I've mentioned, there are 2 main drivers that could drive leukemia. One of them is a hit that cause an element that controls the DNA and gene expression, such as transcription factors or oncogenic translocations that occur in the DNA level and lead to expression of oncogenes. The other one might hit a signaling molecule, such as a surface marker or a downstream of a pathway, and these mutations are usually activating a mutation, meaning the pathway is always active, and these enhance the survival of leukemia and reduce the sensitivity to treatment. For the last 40 to 50 years, the chemotherapy for AML did not change much, and it's made of 2 main blocks. It starts with induction therapy, or called 7+3, and a second one followed by consolidation or targeted therapy. The induction stage aims to debulk the patient from the leukemioblast, and this contains DNA damaging agents, and in order to reduce the leukemioblast from the periphery, which gives room for the second targeted therapy, where here the doctors usually give subscribe depending on the subcases or subtypes, small molecules inhibitors that can inhibit specific pathway after the debulking stage happened. These 2 main phases of therapy, which is the induction and consolidation therapy, are usually the current one which is used in the clinic, and the project that 2 projects that I developed, we believe that they could aid at the second-line treatment, either constituting in the consolidation targeted phase or in the one following after. So the first project was one of the projects was to target a transcription factors and which is difficult to target usually with small molecules inhibitor. And the other project was to target or to interfere with the expression of one of the microRNA, and which have more global gene expression, which also we can interfere with the survival of leukemic cells. So I will start first by talking in general about the platform which I have used. So we used the nanoassembler, and in both projects we encapsulated therapeutic oligonucleotides, either microRNA mimics or siRNA, chemically modified siRNA, so basically short double-stranded RNA. And I used the lipid mixture from Precision Nanomedicine and also from handmade one, but in both cases, it's mainly depends on the DLN MC3 DMA lipid. They formulated nanoparticles. I aim to have them between 50 to 70 nanometers, around 60 nanometers. And this is very important parameter for us that I will follow later. So these nanoparticles, after we tested them in different models, in vitro and in vivo, we found that they are mainly uptaken by the cells with active endocytosis, with involvement of a clathrin endocytosis involvement. So we are targeting leukemia in children, and leukemia is the disease, or myeloid leukemia is the disease of the bone marrow, and the bone marrow is highly perfused, and the endothelial cells of that is very fenestrated, which reassemble the liver. And yes, it is the sanctuary of the leukemic cells, but these fenestrated vessels is important for the bone marrow function. So it allows the nutrient to come in and mature cells to go out. But what also means it's our entry of the bone marrow. So we can use them to target our nanoparticles to go inside the bone marrow and hit the leukemia where they are hidden. To do so, or to know if the nanoparticles we generated, are they able to enter the leukemic reservoir, I developed a click reaction base to decorate the surface of the nanoparticles. So what I did, I incorporated one of the big moieties, which terminated with azide, and then I coupled that with a simple click reaction with alkene coupled to a fluorophore. I used near IR dye, but this system also can be used, or this approach can be used to decorate the nanoparticles with any other ligand, such as antibody or another fluorophore. So this can be generalized to different applications. So after generating the nanoparticles, which are labeled with a dye that allow us to track it in vivo, we injected these nanoparticles with IV into immunocompromised mice. And we tracked the nanoparticle. Up to 3-4 days, we did not see massive change in the PK of the IVs, meaning these nanoparticles has good retention and high PK in mice for a short period following one injection. And if we do systemic treatment for the mice by pre-treating them to saturate the livers of the mice, then injecting the nanoparticles, which are labeled, we were able to track these nanoparticles to a certain extent in the bone marrow of the femur and tibia. We found some even on the brain, spleen, liver, and other organs. So here I would like to mention that the uptake of the nanoparticles is very ubiquitous, and it's not really specific until we go for specification, but as proof of concept that it has very good biodistribution and bioavailability in vivo. Now, I will focus on the first project, where we aim to develop allostotoxic therapy for AML treatment. So, and that's by retrieving one of the microRNA, which is called miR193P. So one major obstacles that face the clinic now is that by the end of the treatment, the patient suffer from severe side effect of the chemotherapy, because this chemotherapy will target leukemia as well as will target the hematopoietic stem cells, the long-term stem cells, and this will interfere with the normal regeneration of the bone marrow. So we aim to develop or to find a target that allow us to cure leukemia, but also at the same time to save the regenerating capacity of the bone marrow by not harming the long-term stem cells. from previous data we had in the lab with different atlases we published, we found that microRNA 193b is a tumor suppressor microRNA, and this microRNA is repressed in acute myeloid leukemia, and it is also has low expression in the long-term stem cells and the hematopoietic stem cells, and it has higher expression in mature cells. So, and this low expression of microRNA miR193b is associated with poor overall survival, with poor clinical outcomes. And so basically patients who have higher expression of this microRNA, they do much better clinically. So we aim to retrieve the expression of this microRNA, or to bring this microRNA expression back. Now, one major advantage of using such a system, we know that one microRNA can orchestrate a global gene expression of multiple genes. And one of the pathways that we found to be targeted with this microRNA was the MAB-ERK pathway. And we do know that the MAB-ERK pathway from our in silico data, that this pathway is silent and inactive on the long-term stem cells, the healthy hematopoietic long-term stem cells, and it is active in mature progenitor and in differentiated cells. And we know that this microRNA is also not active or not present in our leukemia. What does this mean? If we express the miR193b in leukemic cells, this might clear leukemic blast, but this will not interfere with the hematopoietic stem cells. One important note and advantage of using this microRNA is that the MAB-ERK pathway, where several members of this pathway, here highlighted in yellow, are frequently mutated in pediatric patients with AML. And these mutations usually are activating mutations, gain of function, and they are associated with very poor clinical outcomes. So here we are hitting a pathway which is a resistance pathway or survival pathway for these patients. So we planned an experiment to validate that interfering with the hematopoietic system, let's say, or treatment or retrieving the functionality of miR193b will not harm the normal hematopoiesis. So to do so, we have microRNA miR193b mimics and control mimics, and then we encapsulated those into the nanoparticles, and then we treated immunocompetent mice. We applied 2 weeks regimen of treatment, and we sacrificed the mice at the end of the treatment. During the treatment period, mice tolerated the treatment well. We did not see any effect on the movement of the mice, no effect on the blood count. Everything was within the expected parameters. And then when we took out the mice and we scored the number of the LSK cells, or they are the lineage negative stem cells, we found that there is no difference in the 2 treatment groups, meaning bringing back the functions of miR193b with this treatment did not affect the number of stem cells or the healthy stem cells which are retrieved from those mice. We went further and we investigating even if the frequency of each population within those stem cells is affected or not. And what we found that within this LSK or within those within those hematopoietic healthy stem cells, the long-term HSCs, hematopoietic stem cells, and the short-term primed progenitors, MBBs 150 negative, those are also not affected. So not only the total number, but also the distribution and frequency of each stem cells compartment remained intact. What does this mean? This means if we treat the patient which have leukemia with this microRNA mimics, we are able to interfere with the leukemic blast, and we may interfere with the mature cells, because we also found destruction of normal blood cells, mature B cells and T cells and monocyte, but the hematopoietic stem cells and the healthy stem cells are intact, which means by the end of the treatment, the patient will not have severe effect of dysregulated hematopoiesis. So the bone marrow can regenerate again. Now we move to validate the anti-leukemic effect of these nanoparticles that encapsulate miR193b in vivo. And here we relied on a BDX model So it's not a cell line. So this is AML BDXs. We take the cells from patients and we inject them into immunocompromised mice, and we go through different serial transplantation to have a stable BDXs. These BDXs are better modeled than cell lines because they represent the hierarchy and the clonal complexity which are found in AML patients. So after following the treatment, we choose 4 different BDXs. They represent the major subgroups of children leukemia, some with poor prognosis, others with favorable and good prognosis. And what we found that the treatment was striking astonishing for all BDXs, which means retrieving the functionality of miR193B, delayed the leukemia expansion, and we even have complete clearance of leukemia from 2 mice in one of the BDXs or in one of the experiments. It was a complete remission. So what does this mean? It means by the end of this project, we were able to retrieve the functions of a repressed microRNA. And by bringing that, we are able to interfere with the global gene expression that enhance the survival of leukemic cells. And by doing so and applying this treatment, we were able to clear or to reduce the leukemic bulk and to enhance the survival of the mice without affecting the healthy stem cells. And this was the first project. The second project, which was more related to targeted therapy, which relied on targeting a translocation which found in pediatric patients. And here I focused on the translocation 8021, which lead to where we have 2 chromosomes, chromosome 8 and chromosome 21 fused together, and we have an oncoprotein which is called AML-ITO or RONX1-ITO. And what this oncogenic protein is doing is interfering with the transcriptional activity of RONX1. And RONX1 is a main hematopoietic transcription factor and regulator. So now we have a new translocation or new oncoprotein that compete with the wild type transcriptional activity of RONX1, and it dysregulates hematopoiesis. And this leads to changes in the self-renewal, enhance the self-renewal of the cells, and it blocks differentiation. And by this, it makes leukemia. So these type of translocations are very targeted, so meaning they are very specific and only expressed in leukemic cells and leukemic blast, and they are initiating events, meaning they are the first hit that happen in the patient that lead to myeloid leukemia. Having these translocations is very advantageous because they are only expressed, as I said, in the leukemic cells. And what does this mean? It means we can rely on the sRNA, for example, or antisense to target this translocation if this translocation has a unique breakpoint, such as AML-ITO. So in case of RONX1-ITO, we designed sRNA that target the translocation, which is expressed in all patients on the same manner. So there is no difference in the sequencing of this sRNA. First step I did was to introduce different chemical modifications, sugar ribose modifications and phosphate backbones. And what this modification does, it enhances the potency of the sRNA. So I used here, I followed a similar approach to what usually Alnylam or Ionis are following in doing modification for the antisense that they use in their antisense therapeutics. We validated that these modifications are really potent and enhance the potency of the sRNA. And then following that, we encapsulated this modified sRNA as well as a control sRNA into the nanoparticles, and we treated 8021 cell lines, or let's say cell lines for that to express this translocation. And the treatment was very potent, where we found massive reduction in the translocation at the protein and expression level and RNA within 24 hours. And this was very long lasting, that lasted for over 2 weeks following one single treatment. So one single treatment provides us a knockdown or reduction and changes in the transcriptional activity for more than 2 weeks. Now, this massive reduction or depletion of the protein, of the oncoprotein, is associated with the great changes in the genomic level because this is a transcription factor. So we have a great modulation of or depletion of RONX1-ITO transcriptional activity and epigenetic activity and gain and retrieval of RONX1 activity. And that's by controlling different genes. Now, the consequences phenotypically of this meant that the cells, they lost their capacity to perform colony. So we saw massive reduction in colony formation, say, of the cells treated. with these nanoparticles, as well as we introduced a cytostatic phenotype, cell cycle arrest, and senescence, so the cells cannot regenerate anymore. This good phenotype, let's say, for leukemia that block leukemia at cytostatic phase, led us to move forward and test if these nanoparticles are also effective in more complex system. And here we applied the same treatment for AML blast. So this is cells taken directly from patients and cultured on MSC feeder. So we have a co-culture system. And then we applied the treatment and we analyzed after 1 week the data. And what we found that the nanoparticles treatment is also effective in AML blast. And these are very complex cells. They represent different clones taken from the patient. And this system on the feeder layer allow us to study or to recapitulate the leukemic cells and leukemic niche interaction. We found massive depletion as well as in the transcriptional level, as well as in the protein, down regulation of the downstream target of AML-ETO, and we also observed the induction of cellular senescence. So meaning these nanoparticles is also effective in much more complex systems ex vivo. Now we validated this. We moved in vivo to see the on-target activity. So we established a xenotransplantation model by intrahepatic injection of newborn mice with leukemic cells. And this is the only model where it allows the engraftment of this type of translocations or this type of leukemia. And then we treated adult mice. And what we found that when we take the cells, the remaining cells, or when we sacrifice the mice, which were treated with nanoparticles, we saw on-target activity where cells obtained from treated mice had lower expression or lower level of AML-ETO and downregulation of the downstream targets of AML-ETO. Now, this led to enhanced survival, of course, in remission in one of the mice. And interestingly, even though these mice came into a relapse, when we took these cells and we tried to do ex vivo experiment on them, these cells were senescent. These cells did not grow in vitro, but also did not form any colonies. Meaning, yes, some mice came to relapse after this treatment, but these cells were not able to form any colonies, and this led us to do a more complex experiment to see the effect of the treatment on the leukemic stem cells, not only the leukemic blast. And for this, we did another experiment where we injected the leukemic cells into mice, followed by treatment of adult mice. Then we took the cells from the adult mice and then we transplanted them again into new recipient, and we just monitored without any further treatment. And what we found was a striking finding that only 50% of these mice, which were not treated at all, only transplanted with cells pre-treated before, had 50% of them did not develop any leukemia, and the other 50% have complete remission after 1 year. What does this mean? It means that using such a treatment will reduce the leukemic stem cells and will reduce the LIC number or the leukemia initiating cells. And this is something very important because it means if we apply such a treatment for a patient that expresses LONX1-ETO, then we can prevent relapse of that patient or we will have the relapse coming at very later stage. However, again, some mice had relapse. And we took these cells from those mice and we looked at them, what do they express? We found them primed for differentiation. They are already differentiated cells, and these cells could not grow in vitro for a long time. But interestingly, if you look to those cells that we took from relapsed mice and we applied a new treatment, we found that they are still dependent on AML-ETO, meaning they did not develop any resistance. And this is very important because it means if we have a relapsed patient after our treatment, we can apply the same treatment. And this is often not the case with the current chemotherapies because the cells in our case did not develop a resistance and they were still sensitive to the same treatment. So this was the conclusion of the final or the last project. Now, to bring this together into conclusion, we believe that this interfering with these two pathways, the transcriptional pathway, let's say where we have retrieve the functions of one of the microRNA of miR193B. This could be applied to more generic subgroups of AML, because this is a more dependency is for survival for different subgroups of AML, while we have also the specificity of translocation cases where we have such in such case of 8021, we can also interfere with the long-term survival, and we can prevent relapse and delay relapse, and we can apply the same treatment. There are still different questions remain. Of course, we don't know. There are still rooms for PK improvement. There are rooms to maybe combine these two treatments together, where we have one that induces apoptosis and debulk, such as retrieving of a reversal microRNA, while the other one can reduce and affect the leukemic stem cells on the long term, such as AML-ITO. We still need to do and see what is the effect of this on the immune system in a more complex setting, but this is all for the future to be done. So this brings me for the conclusion of my presentation. And here I would like to thank my current supervisor or head, Professor Klusman, who is, as mentioned, he is the head of a pediatric clinic at Goethe University Hospital, and my former PhD supervisor, Olaf Heidenreich, who is now a group leader in the Brexis Maxima Center, and all the universities and funders that I have been working for the last 7-8 years. And thank you very much for listening, and I am welcoming any questions.
Thank you very much for the presentation today. It was certainly very interesting. I'm sure a lot of people found it interesting too. So yeah, we look forward to seeing everyone at the next PNI Teatime Webinar Series. So thank you very much.